Have you ever wondered how the body transforms those extra helpings of pasta or that second slice of cake into stored energy? The answer lies in a fascinating process known as fatty acid synthesis. This process is the body's way of converting excess dietary carbohydrates and amino acids into fatty acids, which are then stored as triglycerides or TG. Fatty acid synthesis primarily takes place in the liver, kidney, adipose tissue, and lactating mammary gland. The biosynthesis pathway occurs in the cytosol of the cell, with acetyl-CoA providing the carbon and NADPH supplying reducing equivalents. The process kicks off with the transport of acetyl-CoA into the cytosol. Acetyl-CoA, produced by the oxidation of pyruvate fatty acids, amino acids, and ketone bodies, is formed inside the mitochondria. However, it cannot exit freely and thus comes outside in the form of citrate. The next step is the carboxylation of acetyl-CoA, where it is converted to malonyl-CoA. This conversion is facilitated by the enzyme acetyl-CoA carboxylase and is considered the rate-limiting step of fatty acid synthesis. The final stage of fatty acid synthesis involves the assembly of fatty acids, a process facilitated by an intricate enzyme known as the fatty acid synthase complex. This complex is a dimer, signifying it is made up of two identical units, each composed of 7 unique enzymes. The synthesis pathway of fatty is initiated by the carboxylation of acetyl-CoA to malonyl-CoA. This is then followed by a sequence of reduction, dehydration, and further reduction steps, resulting in the elongation of the carbon chain. Each cycle of this process adds 2 carbons to the fatty acid chain until the final molecule is synthesized. The metabolic pathways of these fatty acids are diverse and crucial for the body's energy homeostasis. They can be metabolized to produce ATP, which is the body's main energy currency, through the process of beta oxidation. They can also be converted into other bioactive molecules like eicosanoids and prostaglandins, which play pivotal roles in inflammation and pain response. Importantly, this dimer only becomes active when the 2 units are paired, leading to the efficient production of fatty acids. Interestingly, this complex operation is controlled and coordinated by a single gene, ensuring a synchronized operation among all the enzymes involved in this process. The regulation of this process is twofold. In the short term, it is controlled by allosteric or metabolic regulation and the covalent modification of the enzyme. Long-term control is managed by the acetyl-CoA carboxylase, which in turn is regulated by insulin and glucagon. Other factors such as dietary intake, NADPH, and citrate also play a part in this regulation. In summary, fatty acid synthesis is a complex yet efficient process that the body employs to convert excess nutrients into stored energy. It involves the transport of acetyl-CoA into the cytosol, its conversion to malonyl-CoA, and the assembly of fatty acids. This process is regulated both in the short and long term, ensuring an efficient and well-coordinated production of fatty acids. So, the next time you indulge in that extra slice of cake, remember the intricate process that allows your body to store it for energy later.